So today I will start with the topic of DNA replication. I am Qureshi Sahil. So I'll start with translation. So it is the pathway of protein synthesis, and it is the language of the nucleotide sequence on the mRNA. It is translated into the language of the amino acid sequence. So the mRNA is translated from its 5' end to its 3' end, producing a protein synthesized from its amino terminal end to its carboxyl terminal end. So the components required for translation are amino acids, transfer RNA, messenger RNA, amino acyl tRNA synthetase, functionally competent ribosome, protein factors ATP and GTP as a source of energy. So the steps of protein synthesis. The process of protein synthesis is divided into three stages, that are initiation, elongation, termination. So initiation of the protein synthesis involves the assembly of the components of the translation system before the peptide bond formation occurs. So these components includes two ribosomal subunits, mRNA, amino acyl tRNA, specified by the codons in the message, GTP, and initiation factors that facilitate the assembly of this initiation complex. So in prokaryotes, three initiation factors are known, that are 1, 2 and 3, while in eukaryotes there are at least 9, designated as E initiation factor to indicate the eukaryotic origin. So DNA replication, so it is a semi-conservative process. So parental strands are not degraded. Base pairing allows each strand to serve as a template for a new strand. So new duplex is half parent template and half new DNA. So this is the structure of ribosome with light subunit and heavy subunit. A and P sites on ribosome. So the ribosome has two binding sites for tRNA molecules, each of which extends over both subunits. Together they cover the neighboring codons during translation. The A site binds an incoming amino acyl tRNA as directed by the codon currently occupying the site. This codon specifies the next amino acid to be added to the growing peptide chain. So the P site codon is occupied by the peptidyl tRNA. This tRNA carries the chain of amino acids that has already been synthesized. An E site is also there, that is occupied by the empty tRNA that is about to exit the ribosome. So primers. RNA primers are synthesized by primase. It serves as a starter sequence for DNA polymerase 3. Only one RNA primer required for the leading strand. RNA primers for the lagging strand depends on the number of Okazaki fragments. RNA primer has a free 3-OH group to which the first nucleotide is bound. So this is the structure of transfer RNA. Initiation. So the small ribosomal subunit binds to the initiation factor 3. The small subunit initiation factor 3 complex binds to the mRNA. Specifically, it binds to the sequence AGG, known as the Shine-Delgarno sequence, which is found in all prokaryotic mRNAs. Meanwhile, the fMET tRNA binds to the initiation factor 2, which promotes binding of the tRNA to the start codon. The small subunit initiation factor 3 complex scans along the mRNA until it encounters the start codon. The tRNA IF2 complex also binds to the start codon. This complex of the small ribosomal subunit IF3 initiator tRNA and IF2 is called the initiation complex. So this is the unwinding of DNA. When the ribosome is assembled, two tRNA binding sites are created. These are designated P and A. So P stands for peptidyl, A stands for amino acyl. The initiator tRNA is in the P site and the A site will be filled by the tRNA with the anticodon that is complementary to the codon next to the start. So in this case it is the tRNA that binds proline. So when the second tRNA base pairs with the appropriate codon in the mRNA, an enzyme called peptidyl transferase catalyzes the formation of peptide bond between the two amino acids present. While breaking the bond between fMET and its tRNA, This activity is intrinsic to the 23S rRNA found in the large subunit. Since the rRNA catalyzes this process, it is referred to as the ribozyme elongation. So at this point, the whole ribosome shifts over one codon. This shift requires several elongation factors and energy from the hydrolysis of GTP. The result of the shift is that the uncharged tRNA that was in the P site is ejected, and the tRNA that was in the A site is now in the P site. So the A site is free to accept the tRNA molecule with the appropriate anticodon for the next codon in the mRNA. So the next tRNA base pairs with the next codon, and peptidyl transferase catalyzes the formation of a peptide bond between the new amino acid and the growing peptide chain. So once again the ribosome shifts over so that the uncharged tRNA is expelled, and the tRNA with the peptide chain occupies the P site. So this is why this site is called as peptidyl site. After the shift, it contains the tRNA with the growing peptide chain. So the other site will accept a tRNA with an amino acid, hence the name amino acyl site. The process of shifting and peptide bond formation continues over and over until a termination codon is encountered. So the elongation process is fairly rapid, with prokaryotic ribosomes able to add 15 amino acids to the growing polypeptide every second. The process is also relatively error free. Only one mistake is made every 10,000 amino acids. For a large protein of 1000 amino acids, that would mean one wrong amino acid is in every 10 polypeptides. So this is the process of elongation. Termination. So when a termination codon enters the A site, translation is stopped. This is because there is no tRNA with an anticodon that is complementary to any of the stop codons. The release factor causes the translation complex to fall apart and cleaves the polypeptide from the final tRNA. The polypeptide product is now free to the function in the cell. The mRNA molecule is now available to be translated again. So very often more than one ribosome will translate a single mRNA at the same time. One ribosome will initiate translation, and after it moves down the mRNA a bit, another ribosome will initiate, then another, and so on. So the structure consisting of multiple ribosomes translating a single mRNA molecule is called as polysome. So eventually the mRNA is degraded and the translation of that particular message will cease. So this is the termination process. Eukaryotic translation. So eukaryotic translation in every similar overall to prokaryotic translation. There are a few noticeable differences. So these differences includes the following. Eukaryotic mRNAs do not contain a Shine-Dalgarno sequence, and eukaryotes do not use formyl methionine as the first amino acid in every polypeptide. Ordinary methionine is used. Inhibitors of protein synthesis. So the tetracyclines, like they block bacterial translation by binding reversibly to the 30S subunit and distorting it in such a way that the anticodons of the charged tRNAs cannot align properly with the codon of the mRNA. Puromycin structurally binds to the amino acyl tRNA and becomes incorporated into the growing peptide chain, thus causing inhibition of the further elongation. So chloramphenicol inhibits prokaryotic peptidyl transferase. Clindamycin and erythromycin bind irreversibly to a site on the 50S subunit of the bacterial ribosome, thus inhibit translocation. Diphtheria toxin inactivates the eukaryotic elongation factors, thus prevents translocation. Post-translational modifications. So the newly synthesized protein is modified to become functionally active. The various post-translational modifications are as follows: trimming, covalent alterations, phosphorylation, glycosylation, hydroxylation, gamma carboxylation, isoperenylation, protein degradation. So post-translation modifications. Trimming removes excess amino acids. Phosphorylation may activate or inactivate the protein. Glycosylation targets a protein to become a part of the plasma membrane or lysosomes or be secreted out of the cell. Hydroxylation, such as seen in collagen, is required for acquiring the three-dimensional structure and for imparting strength. Defective proteins or destined for turnover are marked for destruction by attachment of a ubiquitin protein. Proteins marked in this way are degraded by a cellular component known as the proteasome. Translation and the summary are given in points. Translation is the synthesis of a polypeptide using the information encoded in an mRNA molecule, and this process involves mRNA, tRNA and ribosomes. So this is the whole summary. So primer removal and nick sealing. So primers are removed by DNA polymerase 1 by replacing ribonucleotides with deoxyribonucleotides. So nicks are sealed by DNA ligase. Multiple primers on the lagging strand while single primer on the leading strand. So proofreading and editing. 1000 bases per second are equal to lots of typos. DNA polymerase 1 used in proofreadings and corrects typos. It repairs mismatched bases, removes abnormal bases, it repairs damages throughout the life. It reduces error rate from 1 in 10,000 to 1 in 100 million bases. Termination of replication. So this process is completed in about 30 minutes. A replication rate of 3 into 10 to the power 5 base per minute base pairs per minute in prokaryotes. So the entire mammalian genome replicates in approximately 9 hours. The average period required for the formation of a tetraploid genome from a diploid genome in a replicating cell. This process is completed in about 30 minutes, as we have discussed. Reconstitution of chromatin structure. So chromatin structure must be reformed after replication. Newly replicated DNA is rapidly assembled into nucleosomes, and the pre-existing and newly assembled histone octamers are randomly distributed to each arm of the replication fork. So DNA synthesis and the cell cycle. So in animal cells, including human cells, the replication of DNA genome occurs only during the synthetic or S phase. So this is usually temporarily separated from the mitotic phase by non-synthetic period referred to as gap 1 G1 and gap 2 G2 occurring before and after the S phase respectively. So the cell prepares for DNA synthesis in G1 and for mitosis in G2. Telomeres. In eukaryotic replication, following removal of RNA primer from the 5' end of lagging strand, a gap is left. So this gap exposes DNA strand to attack of 5' exonucleases. This problem is overcome by telomerase. So mechanism of action of telomerase. So the enzyme synthesizes TTAGGG and repeats onto the telomere sequences using an internal RNA template. Okay, so telomere acts like a reverse transcriptase. It recognizes 3' end of the telomere. Based on the RNA component, a small DNA strand is synthesized. So different level of activity in different cells. So high in stem cells and cancers, activity lost in old age, potential target for newer anti-cancer drugs. So this is the summary of replication. Thank you.